Before we start, here's a little bit information about Flagship, about our story and our capabilities. We at Flagship, even so we are known as a service company, have made great strides to develop our own pathology AI technology. Over the last 8 years, we developed the technology to solve the most challenging tissue analysis problems across the entire pharma industry. Flagship is manufacturer independent, and we can use the best of breed commercially available products. We actually started out using the Aperio platform and image analysis tools, Visifarm and others, but quickly realized that the available commercial image analysis tools do not meet the needs of our clients. So we started to build our own image analysis tools on the Aperio platform. Growing our capabilities and business, we then realized that the available commercial image analysis platforms are not robust and scalable enough for a high throughput operation. So we started to build our own platform. We have been in a unique position to build the best technology and become the technology leader. We have a unique multidisciplinary team of pathologists, biologists, analysts, and our histology lab working together with our engineers to find the best solutions. We are probably the largest team in the world dedicated to computational tissue analysis. We have 2 facilities next to each other in Westminster, Colorado, about 13,000 square feet for our pharma services and our CAP-certified IHC lab, and about 6,000 square feet for our software development and IT. We have built a scalable, high-performance image analysis platform for a high-throughput operation. We designed an innovative system that puts flexible, high-throughput image analysis at the heart of it. Our analysis is defined in form of modular processing pipelines that can be easily assembled to meet the needs of new applications. We are building heavily on computer graphics, providing ultra-fast slide viewing and a very flexible access to our rich information tissue data. The different stains and analysis results can be viewed as different layers, like in Photoshop. On the back end, we have actually built a high-performance computing cluster, HPC, a supercomputer with many CPUs, GPUs, a lot of RAM and shared disks. The core of our image analysis is whole slide cell-based tissue analysis. While today's tissue image analysis challenges are focused on immuno-oncology, which is a sweet spot for our technology as it's too complex for human interpretation using just a microscope and also requires tissue context that cannot be provided by other technologies like next-gen sequencing. We also create tissue image analysis tools for many different applications, including rare diseases. Muscular dystrophy is probably the one we are best known for. Our toolbox uses different machine learning approaches, including those we are talking about today. We work on real-world problems across the pharma and biotech industry. Our technology is validated by pathologists on hundreds of studies and thousands of slides. In the last 5 years, we have done more than 500 projects with 6 out of 10 largest pharmaceutical companies and 300 studies exploring clinical trial endpoints and target validation hypotheses. Over the years, we developed a comprehensive patent portfolio with currently 12 patents granted and 18 pending. In this second part of the Pathology AI presentation, we talk about our Pathology AI system and how the rich information data for tissue that we provide enables healthcare big data for pathology. We introduce Pathology AI step by step, starting with an introduction to pathology, going to digital pathology, and then adding machine learning to create a pathology AI system. We are going to present a smart system design with the pathologist playing a vital role in the center of the pathology AI system, and a patient type-based machine learning approach that allows to overcome a key problem in pathology AI, the variations between patients. To illustrate our concepts, we will provide a demo of our pathology AI system. Pathology is a discipline of diagnosing a disease mostly through analysis of tissue, cell, and body fluid samples. Histopathology refers to the microscopic examination of tissue by a pathologist. The examination starts with a biopsy. Chemical fixatives are used to preserve the tissue from degradation until the specimen gets to the histology lab. The most common fixative for light microscopy is 10% neutral buffered formalin. The histology lab processes, embeds, sections, and stains the specimen. Tissue processing removes the water from the tissue and replaces it with a medium that solidifies to create tissue blocks. The most common medium for light microscopy is paraffin wax. Formalin-fixed, paraffin-embedded FFPE tissue can be stored at room temperature for decades. The tissue blocks are cut into 4 micrometer thick tissue sections using a microtome and are then mounted on a glass microscope slide. Staining is used to give contrast to the otherwise transparent tissue section and to highlight particular features of interest. The most common stain for light microscopy is hematoxylin and eosin, H and E. Hematoxylin stains the nuclei blue, eosin stains the cytoplasm pink. Immunohistochemistry, IHC, uses antibodies to visualize specific proteins and specific cell compartments. IHC often uses DAB to stain the protein in question brown and hematoxylin as a counterstain to visualize the cells. In light microscopy, the absorption of light by DAB staining, which is measured as optical density, is proportional to the amount of proteins. To be able to measure the amount of proteins, appropriate protocols and controls need to be used that control the important variables all the way from the biopsy to the staining. The histology slides are examined under a microscope by a pathologist to determine a medical diagnosis. The performance of pathology is controlled using proficiency training and statistical comparison of lab results to the incident rate in the general population. Histology labs are regulated by CLIA, Clinical Laboratory Improvement Amendments from '88, or CAP, the College of American Pathologists. Histology labs use Laboratory Information Systems, LIS, to manage their workflow. Histopathology has several quite distinct subspecialties, which correlate to the different tissue types, like prostate or breast. Throughout this presentation, we are going to use breast cancer for illustration purposes. Typically, H&E slides are used to determine the diagnosis, like cancer, non-cancer, and a panel of IHC slides are used to determine the right treatment option, requiring an assessment of the protein expressions on the slides in form of a score. Note that there are many different tests in pathology, characterized by tissue type and stain, and each of them only reports a single data point per slide, a diagnosis or a score. Going digital, digital pathology allows to scan the slides and replaces the microscope with a computer monitor. Digital pathology by itself only provides the convenience of dealing with images instead of glass slides. Digital pathology equipment includes slide scanners and a sort of packed picture archiving and communication system that allows to manage, view, and analyze the digital slides. Digital pathology manufacturers are regulated by the FDA. We propose the design of a pathologist-centric pathology AI system, where the tissue analysis is semantically segregated into cell detection, cell classification, and measurements, and the pathologist provides his expertise at the appropriate level. Even so, it might be nice and achievable for some applications, a pathology AI system does not need to be completely automated and locked down, as long as the proper controls are in place. Problems with cell detection will have a huge impact on the analysis. Optimizing the cell detection when needed can only improve the analysis. Cell classification is where the pathologist can bring in his expertise, while relying on a pre-trained system when a pathologist can train the system on the fly. The proper controls are provided by the pathologist, who makes sure that the cell detection and cell classification provides the right results. With the cell detection and cell classification out of the way, all the measurements that provide the data are then just simple computer algorithms that can be verified easily during software verification. Using a pathology AI system now allows us to capture all the information that is in the tissue section, not just a single score per slide. Pathology AI provides rich information data for tissue in form of tissue type specific cell distributions with up to 1 million cells per slide with over 1,000 features per cell. Let's see first a demo before we go into further details. Pathology AI means to provide pathologists with the right tools. We want to illustrate the right use of machine learning for the proper classification of cells, an important and difficult step in computational tissue analysis. Here we show the quantification of IHC-PR staining in breast tissue, which requires the identification of tumor cells versus stroma cells for the scoring. Even so, this seems to be an easy example. It is an excellent example for the evaluation of machine learning, as there is only minimum information available to learn from. Only a single stain, only a single cell compartment, the nucleus. The pathologists provide their unique expertise in a convenient way by providing example regions for the different tissue types to then train the system on the fly. Here we outline one region for tumor cells and one region for stroma cells. The system provides the results quickly, in the order of seconds. The system displays all detected cells, typically between 500,000 and 1 million for a whole slide, with different colors for the different tissue types. Here we display tumor cells in red and stroma cells in green. The pathologists need to verify the proper detection of the cells and the proper classification of the cells. When there are cells that have not been classified properly, then those regions are just added to the training. To save pathologists time, the initial training of the system can be performed by a trained analyst. This is an interactive and iterative workflow until the cells on the whole slide have been classified properly. Beyond the scoring, we provide rich information data for tissue, which means tissue type specific cell distributions with over 1,000 biofeatures. The scoring itself, here the measurement of DAB staining on the nuclei and the counting of cells, are best performed by a computer and can be verified properly as part of software verification. Cells represent the biology we are looking at, and therefore provides a perfect abstraction layer for the semantic segregation. Looking at the overlay of the detected cells on the right and the original images on the left, it seems clear that the detected cells contain all the information there is in a slide image, while providing a reduction in data points by a factor of 1,000. Cells are also the basis of existing scoring schemes and rich information data for tissue. In pathology, we are not looking for arbitrary objects like cats or cars that are in an uncontrolled environment. We are looking for cells that have a certain size and that have 3 cell compartments, the nucleus, the cytoplasm, and the membrane, that can only be stained by a small number of different stains that have distinct colors. Stains may be collocated in some cell compartments, but often aren't. Controls are and can be employed to standardize the staining and imaging process. This is especially important when staining intensities are used as measurements, like in the case of immunohistochemistry, and less important when the staining is used to visualize the morphology, like in the case of H&E staining. It is important to include the clinical laboratories when defining and implementing the controls for the different applications. Cell detection itself is only looking at the proper detection of the morphology of a cell, and therefore doesn't need to be that tightly controlled. It is fine to adjust the cell detection algorithm if necessary, even in a validated clinical workflow. There's a lot we know about and can control in pathology, but there are also a lot of different applications in pathology, with different tissue types and different stains, which introduces a lot of variations in the cell detection. We put a lot of effort into developing robust and modular cell detection algorithms based on traditional image analysis that allow our analysts to create high-performance cell detection processing pipelines for the different applications. This is an area where deep learning has achieved major breakthroughs over the last few years, the semantic segmentation of images, and deep learning algorithms could be used instead of traditional image analysis. On the other hand, we have found that cell detection is not really an unsolved computer vision problem in pathology that is in the way of being able to do pathology AI, like the one we encountered in autonomous driving. Designing deep learning network architectures is an art and can be very tricky. Often existing architectures from other computer vision tasks are just reused. But it might help to take a closer look at what we know about and can control in pathology to define a new architecture. Deep learning approaches will require a lot of training data that is quite expensive as it needs to be annotated by pathologists. We actually create that data for free in our demonstrated machine learning workflow, where pathologists verify the proper detection and classification of cells for the whole slide for the entire data sets. If one day we are looking to automate a specific application, we have all the data we need. Over the last 8 years, as we worked through hundreds of studies, we have developed an extensive set of a few thousand biology-motivated features. We call those biofeatures. Basically, we are looking at morphology and staining at the cell level, in particular at the different cell compartments, at cell neighborhood levels, and at the region level. Patient-to-patient variations is a key problem that affects the classification of cells. To distinguish between different cell types, we notice that the same cell type has different characteristics in different patients. Here, in patient 1, tumor cells can be described as dense, large nuclei, and stroma cells as dense, small nuclei. So the characteristic feature to distinguish between tumor and stroma cells for patient 1 would be something like nuclei size. In patient 2, tumor cells can be described as dense small nuclei and stroma cells as sparse small nuclei. So the characteristic feature for patient 2 would be something like nuclear density. On the other hand, if we are looking at a single patient, we notice that there's typically a single or just a few dominant features that allow to distinguish between the different tissue types, like cell size in patient 1 and cell density in patient 2. Also, we notice that there's only little variations in those characteristics across the whole slide. Interestingly, in our example, the stroma cells in patient 1 have the same characteristics as the tumor cells in patient 2, dense small nuclei. So, if we would create a machine learning system that was trained only with patients that have the same tissue type characteristics as patient 1, the system would fail when it encounters a new patient type, like patient 2. This illustrates the bias of machine learning systems that originates from the data used for training. One way to overcome this bias problem is to throw a lot more data at it, and to make sure all patient types are included appropriately in the training data. Often it is relatively easy to build machine learning systems that provide 90% performance, but then it becomes exponentially harder to get to 95% or even 99% performance, as those remaining exceptional cases are hard to come by. And if we would create a machine learning system that was trained with patient 1 and patient 2, the system would have to learn somewhat contradictory data, which would require the use of complex machine learning approaches that need a lot of data for training. Maybe even the creation of new, non-obvious features that somehow allow to distinguish between the different patient types, and it would become quite difficult for a human to understand the underlying model. Looking at those problems, and with the current hype about deep learning, it may seem obvious to use deep learning and to solve those problems by throwing a lot of data at it. Another approach that we have shown in our demo is to create a new classifier for each patient type. So if we encounter a new patient type, like patient 2, we will be able to create the appropriate classifier for it. Basically, we are creating a bank of classifiers for different patient types that can be added to even in production. Limiting the machine learning to a specific patient type simplifies the machine learning problem considerably. Providing excellent classification results with simple machine learning approaches like decision trees and only requiring data from a few regions for training. The training is ultra fast and can be done on the fly in an interactive and iterative workflow. A decision tree based on biology motivated features provide easy interpretable results to biologists and pathologists and a meaningful grouping of patients by patient type. Let's take a closer look at all this by using some machine learning on our example. As we have shown in our demo, training a simple decision tree using a grid search with cross-validation to determine the best hyperparameters automatically on a single slide using all our features identifies a single important feature, the mean nuclear area. Interestingly, this is the same one we observed by eye. This nicely illustrates the meaningfulness and interpretability of the results that can be provided by machine learning. This single feature with a simple threshold provides excellent results over the entire slide with an estimated accuracy of 98%. As anticipated, using the classifier that we trained on patient 1, on patient 2 does not work. This is a good illustration of the bias problem from training data. Interestingly, this suggests that a fully automated machine learning system should include a review by a pathologist to be able to catch these cases. But then it wouldn't be a fully automated system anymore. The training of a simple decision tree with about 10,000 data points that have more than 1,000 features is ultra fast and takes only a few milliseconds. As we pointed out before, to be able to implement the right machine learning methods, a good understanding of the data is required. Cells in the same region typically have very similar characteristics. Therefore, any effective cross-validation needs to be done by segregating data from different cell regions. When we train a classifier for a specific slide, we call that a per-slide classifier. When we train a new classifier for patient 2, using the same hyperparameters and limiting the features to morphology features to make sure the system ignores the DAB staining, identifies a single important feature, the number of cells in a cell neighborhood. Again, this matches nicely what we observed by eye. Again, this single feature with a simple threshold provides excellent results over the entire slide with an estimated accuracy of 95%. When we train a classifier based on multiple slides, we call that an across-slides classifier. As we can see here, the training of a single classifier using the same data from patient 1 and patient 2 requires a more complex decision tree to achieve a similar performance. Rather than one important features, we get a list of multiple important features that still include the mean nuclear area and the number of cells in a cell neighborhood, but with much lower importance factors. That clearly won't lead to the same interpretation that there are two different patient types, one characterized by the nuclear size and the other one characterized by cell density. There are huge variations in the characteristics between patients. Therefore, any effective cross-validation for the training of cross-slides classifiers need to be done primarily by segregating data from different slides. As in a cross-slides classifier will have to learn many different patient types, more complex machine learning approaches are required. We show here our example with a simple decision forest, built up of 100 decision trees using just bootstrapping, but larger and more complex decision forests with randomization of features and thresholds can be built as necessary. One advantage of decision forests is that they still provide a list of important features, like the decision trees, that help with interpretation of the results. We have parallelized the implementation of our decision forest and run our machine learning algorithms as part of our daily workflow on our high-performance computing cluster. Even so, there are a lot more decision trees that have to be trained, and more data from across multiple slides is used for the training. The training of a decision forest can be done in the order of seconds. There are huge variations in the characteristics between patients. Therefore, any effective bootstrapping approach should consider segregating data from different slides. Let's come back to our approach of the per-slide classifiers for different patient types. We have developed different mechanisms to automatically select the best per-slide classifier for a new patient, but the concept can be best illustrated with our very robust manual method. A pathologist simply outlines a representative region for one or more tissue types. As the processing is ultra-fast, we evaluate in a few seconds all different per-slide classifiers against those regions and determine the best fit. When the classification results are then shown to the pathologist, he can choose to use this classifier if the results look good, or simply create a new one for this new patient type. Our approach can be started with just a single slide and a few region outlines. No need to create an extensive training data set that includes all possible patient types. There is no risk of the bias from the training data we talked about before, as each patient is reviewed by a pathologist. The key to the performance of our system is the extensive and appropriate feature set that we have developed over the years and that allows us to create a high-performance per-slide classifier for any slide. Ultimately, all patients are grouped by the per-slide classifiers that fit their tissue characteristics best, and which then can be described by a simple decision tree with biology-motivated features. Rich information data for tissue now consists of cell type specific cell distributions with all those 1,000 biofeatures per cell. Conceptually, this data is very similar to the data obtained from flow cytometry, except with tissue context. Here we show the histograms of the two features we talked about in our example, the mean nuclear area and the number of cells in a cell neighborhood. Pathologists typically provide a single score or interpretation, which is very specific to a given application and its established scoring scheme or interpretation guideline. We are asking a pathologist to take a look at a histology slide under a microscope. That is even not a cleared medical device and that is often not calibrated properly. To assess 500,000 to a million cells that can show considerably heterogeneity across the slide. And then when it comes to simple scoring, we ask him to determine a percentage of cells of a certain cell type that have a staining in a certain cell compartment that is above an absolute staining threshold. This seems to be a very challenging task that leads to high inter- and intra-pathologist variations. Today's biomarkers, mainly driven by Immuno-Oncology, will require the pathologist to deal with more stains, look at distances between cells, and apply more complex scoring schemes. This is becoming impossible. Obviously, we ask for a lot of things any human has a hard time doing, but a computer can do without a fault. Maybe it's time to replace a microscope with the right tools, a pathology AI system, where the pathologists bring in their knowledge about the tissue, but then have a computer count the cells and do the objective measurements and scoring. The use of rich information data provides an accurate and precise scoring with no limitation in the complexity of the scoring scheme. In our example, we show just a continuous score that provides values between 0 and 3.5, where 0, 1, 2, and 3 corresponds to the traditional 0, 1+, 2+, and 3+ scoring. We suggest an effective and efficient validation and verification of pathology AI software by segregating the part that can change from application to application and from patient to patient from just simple computer calculations. The cell detection and cell classification are the parts that depend on the application and the patient and are therefore best verified for each patient by an expert pathologist, providing 100% coverage. The counting of cells and the calculation of all our biofeatures, any other measurement, and the scoring scheme are based on simple computer calculations that can be verified properly as part of software verification, without requiring a specific knowledge about the different applications or patients. In our example, we would be looking at a software verification for the proper calculation of the nucleus area, the mean optical density of the DAB staining on the nucleus area, the mean of the nuclear area in a cell neighborhood, the number of cells in a cell neighborhood, the accumulation of the DAB staining feature for tumor cells only in form of a histogram, and the scoring scheme that calculates a score based on the histogram. People may think that an automated pathology AI system that achieves a performance of 90% outperforms pathologists. But those pathologists are using microscopes, and the part they are not good at would greatly benefit from the use of computers. So, is that the right comparison? How good are pathologists actually? Anecdotally, it is interesting to see how pathologists, like nobody else, can agree with other pathologists. Even so, their scoring or interpretation was different. This probably goes back to the fact that they agree on what they see in the tissue, their expertise. But when it comes to the counting of cells, objective measurements, and scoring, humans are not consistent. Stereology was invented to overcome some of those problems, like heterogeneity. Stereology is not practical in a clinical setting because it takes too much time. But it is the perfect tool when it comes to the performance evaluation of the cell detection and cell classification. It would be impossible to have pathologists outline every cell or even every tumor region on a slide to provide the ground truth for an evaluation. But it is possible to have a pathologist annotate a certain number of randomly placed small regions with a statistical framework that allows to extrapolate the results from those regions to the performance on the entire slide with a provided error margin. Automated pathology AI systems will have a hard time to increase their performance to 95 or even 99%, but that might be required to get close to the performance of a pathologist using the right tools. A key concept we are going to present is that a pathology AI system needs to provide rich information data for tissue to enable healthcare big data for pathology, allowing us to benefit from the power of data science. The ultimate goal of pathology AI is to provide rich information data for tissue to enable healthcare big data for pathology. Therefore, pathology needs to move from a test-specific single score per slide to general-purpose rich information data that is consolidated at the cell level using standardized panels with multiple markers that can be run in a clinical environment. The challenge here is not the technology. The required registration algorithms already exist, but multiplexing on the wet assay side. In particular, trying to put as many markers as possible on a single slide. To push the limits, histology labs need to work together with technology experts, allowing them to use computational tissue analysis to provide objective quantitative measurements to guide the proper development of the wet assays. As those wet assays might first be too complex to be run in every clinical lab, specialty labs like Flagship would be able to offer them nationwide in the proper regulatory environment. The rich information data for tissue now enables healthcare big data for pathology. Using data science, new insights can be obtained by mining the data. New scoring schemes can be obtained by correlating the data to clinical outcome data. Bioinformatics data, information and knowledge bases, like protein ontologies, can be added to provide more meaning to the data, and tissue data can be combined with other rich information data sources like next-gen sequencing, which provides complementary data to tissue. Data science is built on top of the rich information data provided by Pathology AI. Today's open source software community provides a rich ecosystem based on Python that provides the latest state-of-the-art statistical analysis and machine learning tools. And today's cloud computing provides all required resources necessary to meet the most demanding needs for big data applications.